So we have talked about tight junctions. Now let's talk about the next category of junctions, the anchoring junctions. Anchoring junctions connect cytoskeleton of a cell either to the cytoskeleton of its neighboring cell or the ECM or the extracellular matrix. As we have discussed earlier, the plasma membrane does not have any structural strength. It is fluid. Molecules which are present in the plasma membrane, they're basically floating around. So if force is applied on a cell, it is stretched or it is compressed, the cells will not be able to resist any of these. The membrane of these cells will not be able to resist any of these forces. We are going to talk about the stretching forces, for example, or the tensile forces. So if these cells, for example, if this cell was pulled away, it will just simply come off the plasma membrane, or the cell will just conveniently come off if it was not attached to something. So here these anchoring junctions basically anchor the cells in two ways. Either they hook the cell to its adjacent cell or they hook the cell up to the ECM or extracellular matrix. Just hooking up the membranes of two adjacent cells or membrane of the cell to the extracellular matrix is not enough, because again membrane does not have any structural strength. So these anchoring junctions basically are connected to the cytoskeleton or the skeleton of the cell, which can resist the tensile forces. So the function of these anchoring junctions is not just to hold cells in places, but also they have to connect the cells to the cytoplasm of the adjacent cell or the ECM or the extracellular matrix. Let me explain this point to you in a different way. Let's assume that we have a bird cage. The wires of this bird cage are basically the actin filaments. And on top of these wires, we have a mesh. The mesh here basically represents another cytoskeletal element called spectrin. We will talk about cytoskeleton in a much greater detail later on, but here since we are talking about junctions, it is important that we have some basic understanding of a cytoskeleton, because they play a role in junctions, as we will see. On top of this mesh, we put a piece of cloth. The piece of cloth is our plasma membrane. The piece of cloth does not have any structural integrity or structural support in our case, because if we put a piece of cloth on a table, it will become square. If we take that same piece of cloth and put on a ball, it will become a sphere. So this cloth basically is fluid. It is molecules embedded in this piece of cloth can move around. An actual piece of cloth, they would not move around, because the strengths present in the cloth do have structural strength. But for our case, let's just assume this piece of cloth, things can move around in it. These elements are also basically present at the periphery. In the very middle, we have a structure. This is the centrosome, and to the centrosome, we have hollow pipes connected. These hollow pipes basically have the strength. If we were to press on this cage, they will resist the compression forces. If we were to pull on this cage, the actin and spectrin is going to resist some of this tension. But what is the major element that is going to resist tensile forces is these rope-like structures you can see here. These are the intermediate filaments. These intermediate filaments are connected to some of the anchoring junctions. Here, for example, these rivets, or if you will, the titch buttons present on the side of the cell, which will stick to the titch button on the adjacent cell. So if I were to pull on this cell from this side, this titch button will also, which is connected to the cytoskeleton of the adjacent cell, will also pull on that particular cell. So the force will be dissipated in a way. Additionally, some of these titch buttons are present in the lower part of our cage, and these are attached to the extracellular matrix. So not only the force will be dissipated by the by the adjacent cells, but also ECM will also resist, or the extracellular matrix will also resist some of this tensile force. So this is basically how the cytoskeleton is put together. Additionally, I did not show in this case, but there are actin filaments present at the apical site circling in 3 dimensions. They are hooked to a different type of junction. We'll talk about that later on. But those are adherent junctions. We'll talk about them later. But these rivets are basically a type of cell junction which holds 2 cells together. And these rivets at the bottom, they're different type of cell junction that hooks the cell to the basement membrane or the extracellular matrix. So let's continue our discussion. Additionally, just think about it. If we know that our muscle has different fibers, the actin and myosin fibers, and myosin and actin fibers, they can slide against each other. If this was happening inside the cell without actin and myosin fibers attached to adjacent cells or the ECM, they will just simply slide against each other in the cell cytoplasm without generating any force which can be used to do work. So if these fibers, these actin myosin fibers are attached to something, in this case they are attached to a tendon through some of these junctions, anchoring junctions that we will be talking about. When these fibers slide against each other, they can pull on the ECM, which is a tendon, and result would be the bone will move along the joint. So again, the anchoring junctions play a very important role in biology. They are widely distributed in animal tissue. Most abundant tight junctions, most abundant in tissues subjected to severe mechanical stress, for example, muscle, epidermis, etc. There are 2 classes of proteins which are used to make these anchoring junctions. There are intracellular anchor proteins. They form a distinct plaque that connect the cytoskeleton, actin or intermediate filaments to the transmembrane adhesion proteins. So here, for example, these are the intracellular anchor proteins. They are connecting the cytoskeletal elements or cytoskeletal filaments to the transmembrane adhesion proteins. So one type of protein is the anchor protein, and the other is the transmembrane adhesion protein, which is sticking outside the cell, and either it is grabbing the extracellular matrix or it is grabbing hold of a different protein, transmembrane adhesion protein of an adjacent cell. So these are the 2 different type of proteins. There are also 4 different types of anchoring junctions. 2 of them, adherent junctions, desmosomes, hold cells together and are formed by transmembrane adhesion proteins that belong to cadherin family. We'll talk about cadherin family proteins in greater detail subsequently. But for now, I want to mention that there's another class of focal adhesion, another class of anchoring junctions called focal adhesions and hemidesmosomes, which bind cells to extracellular matrix and formed by transmembrane adhesion proteins of integrin family. So again, the cadherin family proteins are the sticky proteins that hold the 2 cells together. They form 2 structures, the adherent junctions and the desmosomes. And the anchoring junctions that anchor cells to the basal lamina or extracellular matrix, they are hemidesmosomes and focal adhesions. These focal adhesions or hemidesmosomes contain a protein which is of the integrin family, whereas these junctions, they contain proteins of cadherin family. Here again is a summary of what I just said. Adherent junctions, they have cadherin proteins, they hold neighboring cells together and they attach to actin filaments. Desmosomes, they also have cadherin proteins. Sticky proteins are cadherin proteins, and they also the extracellular ligand, or they rather than the type of protein they hold of the neighboring cell is also a cadherin protein, and they attach to intermediate filaments. The type of protein, I don't want to get into too many names here, but anyways, desmoglians and desmocollins, we'll talk about them later. Focal adhesions have an integrins, and so do hemidesmosomes. Focal adhesions are attached to extracellular matrix proteins, so are the hemidesmosomes. Focal adhesions have actin filaments, and hemidesmosomes have intermediate filament attachments. So we have just basically talked about in basic structure, basic different types of anchoring junctions. We've talked about 4 of them. We'll look at them in more detail in subsequent module.